So hello everyone, how are you all? I hope you all are fine. So in the last video, we have discussed about some organelles. Those are endoplasmic reticulum, Golgi apparatus, lysosomes, secretory fascicles. Okay. So in today's video, we will be discussing the remaining organelles, and those organelles are nucleus, mitochondria, ribosome. Okay, fine. So in this video, we are discussing about nucleus, mitochondria, ribosomes. So starting with the nucleus. Okay, it's a cell organelle where DNA is generally present. Okay, so a nucleus contains the DNA. Okay, and this DNA is nothing but the genetic material. DNA contains some genes. Okay, and those genes are responsible for encoding proteins, and that proteins gives the unique identity of a particular cell. Okay, suppose this is the GM. Okay, fine. Now, this is the DNA we all know. But there is some particular sequence, suppose from here to there. Okay, this particular sequence is responsible for encoding protein, which gives a unique characteristic, of a cell. So, this particular part is known as gene. Okay, so by this, we can say that the DNA in where specific part which encode for protein and which is responsible for the unique characteristic of a cell, that particular part is known as the gene. Okay, fine. So now, if we see the structure of a nucleus, okay, no, this is a cell, okay, this is the nucleus, and this portion is the nucleolus, okay, and this covering is the nuclear envelope, or we can say as nuclear membrane, okay. This is the nucleolus, okay, and between the nuclear envelope and the nucleolus, the fluid that is present is known as nucleoplasm. Okay, fine. So this is the structure of the nucleolus. So the outer covering is the nuclear envelope, the center part is the nucleolus, and the fluid that is present between the nuclear envelope and the nucleolus is the nucleoplasm. Okay, so in the nucleoplasm, DNA is present. Okay, but in the form of chromosomes. So DNA is being packed condensely in the form of chromosomes. Okay, so the structure of the chromosomes is like this. Okay, like this. Fine. So these are the structures of the chromosomes. Fine. Now, these genes, these genes, how genes gets converted to proteins? We will see that genes first get transcribed mRNA. Okay. Then this mRNA gets translated to proteins. Okay. So in between, there are some subsidiary steps. So we are not discussing it here because it's a complete part of the genetics. But in the physiology part, only how genes get processed to form protein via transcription and translation. Okay. And this overall process is known as central dogma. Okay. This overall process is known as central dogma. And this particular protein is responsible for unique characteristics. Means unique identity. Okay. Or identity. Fine. Now, talking about the nucleolus. Okay. This nucleolus is formed by accumulation of RNA and proteins. Okay. So nucleolus are the accumulation of RNA and proteins. So let's see the process. The DNA that is present in the nucleus, okay, gets converted to RNA. Okay. This RNA is being stored in the nucleolus. Okay. And the rest of the RNA move out through nuclear pore into the cytoplasm. Proteins are present. Cytoplasmic protein. And with the cytoplasmic protein, this RNA gets assembled and forms ribosomes. Okay. These ribosomes have two parts. One is the smaller subunit and the other is the larger subunit. The smaller subunit is the 40S ribosome and the larger subunit is the 60S ribosomes. Okay, fine. So how this RNA gets stored in the nucleus and how this RNA via nuclear force into the cytoplasm gets assembled with the cytoplasmic protein to form ribosomes. Okay, now it's clear to all of you this process. Fine. Now, mathematically, if we see this 40S and 60S lysosomes, it gets 100S lysosomes. Mathematically, if we see. But when we see this smaller unit and the larger unit, when it gets assembled, okay, the smaller unit and the larger unit, when it gets assembled, it gives 70 years, not 100 years. Why is that so? So the concept lies between the centrifugation process. Okay? Centrifugation process. Okay? This 70 years, okay, this S stands for, this S stands for sedimentation. Wait, okay? Or we can say as Swedberg's coefficient. Swedberg's coefficient or unit, whatever maybe it is. Okay. So sedimentation rate means how quickly these units will get sediment by centrifugation. Suppose this smaller subunit gets sediment at 40 years. Okay, we have seen this. And the larger subunit centrifuging, it gets sediment at how much? 60 years. But in total, small plus large, in total, when we have centrifuged it, it sediments at 70 years. Okay, it sediments at 70 years. So it's not mathematically actually, it's by the centrifugation process. It has been seen that the smaller subunit quickly sediments at 40 years, larger subunits little take more time to sediment at 60 years. But when we combine the smaller subunit and the larger subunit, it sediments at 70 years rather than 100 years. So it's a biological process, not the mathematical process. Okay. So now I think there is no confusion between why 60 and 40 years become 70 years, not 100 years. Okay. Now it's clear to all of you. Fine. Okay. So we have discussed about nucleus, nucleolus, ribosomes. Okay. Now the last part that is been remained is the mitochondria. Okay. Mitochondria. So we all know this mitochondria is the powerhouse of a cell. This is the universal definition. We all know this. Mitochondria is known as the powerhouse of a cell. Why it is known as the powerhouse of a cell? Because in mitochondria, generation of ATP takes place. And ATP serves as the energy. It acts as the energy currency for the cell, by which all the cell gets energy and ultimately we get the energy. And when we get the energy, we can able to do many activities. Okay. So mitochondria is the power of a cell because in mitochondria, HD generation takes place. Okay, fine. So how ATP is been synthesized in the mitochondria? Okay, that I have explained in a separate video. So I request you all to watch that. Okay. So I have explained in details how much is required in the physiology. In that much depth, I have explained how in mitochondria ATP generation takes place. Okay. So lastly, if we see the structure of the mitochondria, it has some outer membrane. Okay. Some outer membrane, some inner membrane, and the inner membrane has certain projection which is known as crista. Okay. So this is the outer membrane. This is the inner membrane, and these are the crista, and this is the matrix. Okay. In matrix, there are some enzymes. Okay. These enzymes are known as oxidation phosphorylation enzyme. Enzyme. Okay. So these enzymes are responsible for those biochemical reaction to synthesize ATP. Okay. Fine. So this is the diagram of mitochondria, the structure. Okay. So we have covered all the 10 organelles. Fine. So if you have any doubt, please ask me in the comment box. I will definitely try to sort it out. And thank you.